Welcome to the Leaving Cert Biology channel. This ties hand in hand with our website leavingcertbiology.ie, which is a platform for 5th and 6th year students in Ireland studying Leaving Cert Biology. Of course, we have our TikTok and Instagram page at leavingcertbiology.ie as well, where we offer completely free videos, tips, tricks, exam strategy and trends. And on our website, we have exam paper structure, timing, all of our completely free podcast episodes and so much more. Our aim is to support students all over Ireland. We're going to start off in this video by looking at Unit 1 chapter known as the scientific method. So we're going to start off with Unit 1, the scientific method, which is actually a short chapter but tends to come up as a 20-mark question. We're going to look at the definition for biology, bio meaning living, so this is the study of living things such as plants, animals, microorganisms, don't forget about those. Within biology, there are so many areas that can be studied. So for example, botany is going to be the study of plants, microbiology is the study of bacteria, fungi, viruses and other microorganisms, anatomy is the study of the human body, and zoology is the study of animals. Then we're going to look at the definition for the scientific method. So it's a series of steps used in an investigation to answer questions relating to the world around us. Okay, and we have to talk about the steps of the scientific method in the order in which they occur. That's a common question. Step number 1, and we will go into these in a bit more detail, is making observations using our senses, such as our sight, our hearing, our smell and our taste. From those observations then we can come up with questions that we want answered relating to the world around us. Based on our observations, you are going to form what's called a hypothesis. Please do not use the word guess in your answer when describing or defining hypothesis, and we will give a more accurate definition later on. From your hypothesis then, you are going to test your hypothesis to prove or disprove it, and how you're going to do that is you're going to design and conduct an experiment. There are some features of an experiment that will make it a good one, such as having a control, having only one variable that changes at a time, obviously ensuring that it's safe and that can actually be carried out using the equipment that you have in a lab. After you conduct an experiment then, what you're going to collect from it is step number 4, results. So results, data, is going to be collected during your experiment, and that's what's going to be analysed. Data can take many different forms. Some data can consist of numbers, some will have nothing to do with numbers and could be changes in colour or smell. We will get on to the different types of data later on in the video. Following on from the results then, these results are going to be interpreted and your hypothesis is going to be accepted, rejected or changed, and this is going to form our conclusion. Okay, so our conclusion is going to contain all of that information. Then what's going to happen is scientists obviously document their experiment and their findings as they go along, and they are going to report and publish these findings in what's called scientific journals. Okay, so be careful that your wording in exam paper answers is specific. And then we are going to have number 7. The hypothesis becomes what's known as a theory when it's supported by a large body of evidence. Remember, a theory and a law are not the same thing. A theory has not yet been proven wrong, but it is not 100% proven correct. So a scientific law is a statement based on repeated experimental observations that have proven to be true. So a law is proven 100% to be true. A theory is widely accepted. Definition then for a hypothesis, a possible explanation for an observation which can be tested. A hypothesis can be accepted, rejected or changed. Okay, so be careful, include the words tested and do not include the word guess. For results, results are data or information collected from an experiment. Data can be quantitative or qualitative, and we're going to talk about those now. The way I remember this is quantitative has the letter N in it, N for data containing numbers with an N. Okay, so we know we can collect data such as temperature, the time something takes. So that's our first one. Quantitative data consists of numbers or numerical data, such as time, temperature, all involving numbers. Then our next type of data is going to be qualitative. Qualitative data is not going to consist of numbers. It is going to talk about maybe the physical appearance of something, the smell of something, the colour change of something, your observations not involving numbers. After this, then we need to talk about what's called a control. So a control is really important in an investigation as it acts as a comparison to which the actual experiment can be judged against. It is identical to the test experiment but without the variable that is being tested. Usually in biology, water is a very common control that is used. So it is a part or a feature of having a good experiment. So principles then of good experimentation: they must be carefully planned and designed, carried out in an ethical, safe manner, include a control, and be free from bias. And we're going to talk about ethics in a little bit later on. Obviously, safety is paramount and crucial with regard to carrying out an experiment. And we're going to go into bias in a second. So an experiment should be designed as a fair test. You can only test one variable at a time to ensure a fair test, and so you know what exactly is causing changes in your experiment. A fair and unbiased test is achieved by using a large enough sample size, so ensuring that you're testing on a large or a wide range of people, for example. Number 2 then, a fair test and an unbiased test is achieved by using a random selection of samples. So for example, sampling a wide range of age groups if you're testing on a human population, and gender as well. Okay, ensuring that there's good variation there. Number 3 then is repeating the procedure, also known as replication or replicates. Obviously, the more you repeat a procedure, the more results you get, and the more accurate then your analysis can be. And then number 4, double-blind testing. So double-blind testing, we're going to give the definition for it now in a second. This refers to when neither the person that is doing the testing, so not the patient nor the researcher, knows who is receiving the drug, for example, and who is receiving the placebo. So this avoids bias because nobody knows who's the experimental group and who's the control group. Ethics then refers to whether a conduct is morally right or wrong. Some ethical issues can surround testing on animals or cloning or genetic engineering. They have been some things over the past few years. Then we have limitations of the scientific method. So we have insufficient knowledge. Method of investigation could be inadequate, leading to incorrect results. We could misinterpret the results, changes in our natural world, or accidental discoveries. That is the scientific method. Thanks so much for watching. Please hit the follow button, comment below if you enjoyed it and want to see more, and follow leavingcertbiology.ie.